I'm Katie.
I'm Vinny, and this is Learn Real Good.
Do you remember when we started this podcast?
We wanted to have a song.
We had a jingle,
Yeah.
and we have not attempted it in years.
No.
I wonder if you remember your notes. It's like the NBC notes. Do you remember?
I could try.
1, 2, 3.
Learn real good.
I think I was off.
Well,
Anyway.
That's that song.
That's what the show's been missing all this time. How have you been, Vinny? What's new? What's on your mind?
I'm good. You know what? I just found out we're about to have a heat wave. And I don't like that information.
You know, we try to stray away from conversations about time because
guess
episodes
what?
come out way later.
This heat wave has got me shook.
This is going to be coming out
In November, probably.
November.
Yeah.
Late November.
Yeah, where I'll be grateful for a heat wave.
Would you?
No.
We're fall people.
I love fall and winter. Summer,
October, I'd say, is my favorite weather month. September is still hot, as we currently are feeling.
Yeah, because we are currently in
September.
September.
All right. It's September 3rd, 4 p.m. I said it. Okay, deal with it. It's a different time for you. So what?
Look, I'm not a crazy fan of the heat. I don't do well in it. I don't enjoy it. Give me the cold. I always tell people, I always prefer minus 40 to plus 40.
Yeah, well, I came from Edmonton to Montreal.
Yeah.
And in Edmonton, the summers, as you are annoyed with me talking about this point, perfect summer weather. Mm-hmm. Low, mid-20s.
Yep.
Sunny most of the time, unless it was like a rainstorm.
It's very fun. You cannot say the word Edmonton without immediately following it up with the summers.
The perfect summer weather, in
you
my
cannot
opinion.
say anything about the city of Edmonton without this coming
Winter,
out.
very cold. But I would take it, mostly because of the lack of humidity.
Sure.
Here, it's so warm and humid, and no one has air conditioning. It's like this weird combo.
We didn't need it. We didn't used to need it.
Well,
now
what happened?
Well, I don't know. Maybe the climate's changing.
I don't believe it. Well, I definitely do. Can't joke about that. This isn't a show about climate change, though, or the weather, local weather. Imagine it was a podcast just about local weather.
That would come out 12 weeks later.
Yeah. What it is a show about science.
Science.
Comedy.
Science.
Believe it or not, this is what we think comedy is. And we shoot the breeze.
Check. And then exchange science facts. Facts. And then we have a great guest.
Yes, I can't wait for our guest.
Yes, I'm always excited.
But first, we have to share science facts.
We can't skip a step.
The guests will be irate and angry and gassy if we do not share science
gassy?
facts. How else do they get their science facts?
Gassy?
What?
You said gassy.
Why do you keep saying gassy? Shall I go first?
Sure.
Ladies before gents. Well, Vinny, a few episodes ago, I started very similarly. I'm going to do it the same. What do fire beetles, snakes, and vampire bats have in common?
All delicious.
Gotta take a guess. It's impossible that you're going to get this, but I'm still going to guess.
All right, so what are they? Fire beetles?
Certain snakes. And vampire
bats. Important to the regrowth of a forest after a fire.
That's it. No.
Oh.
They all have this special superpower that is pretty rare, and that is the capability to sense weak radiating heat.
Weak radiating heat? What does that mean?
Well, you can smell, you can see, you can taste. Well,
yeah.
some animals have an additional sense, a sixth sense, if you will, to see dead people, to sense thermal energy.
It says hot people.
Yes.
I have that sense.
The real hot or not is in vampire bats. Now, I bring this up because we've recently found, this is actually a couple years old, but I just found this.
Okay.
Recently-ish. Come on now. That dogs can too.
Dogs can sense heat.
Correct. Thermal energy.
Thermal imaging.
Weak thermal energy. Exactly.
Right.
And so this kind of helps explain why dogs who can't, are lacking in some other senses can still hunt.
Common sense.
Yes, common sense, hearing, comedic sense, and the thermal sense can help them hunt.
Okay, so it's not just their sense of smell.
Correct. Well, speaking of which, though, how many times more sensitive is a dog nose at smelling than ours? Do you know?
2.
Of course, it's more sensitive.
2 times.
100 million.
What? 100 million?
100 million
they
times.
They can detect things 100 million times more sensitively.
Can you imagine what that stinky dumpster we smelled in Belgium, which is
it's our barometer for stinkiness.
For worst smell. Yeah. What a dog.
It was a hot day in Belgium, and we opened a fish market dumpster to throw something out. And to this day we are scarred.
I hate to quibble. It was a cold day.
It was,
but it smelled like a hot day, let me tell you.
Okay.
Anyway, so dogs can sense thermal energy.
Yeah.
And apparently, like, this is, it seemed people were suspicious they might have this ability because of their nose. Having a nose that
why
is
it's cold
moist
and wet?
and cold.
Yeah.
Well, it's not well, maybe it is. But that is a common element amongst some animals, I guess.
So are they going around like, like the predator vision?
Exactly.
Yeah, it's like
a little bit.
Do.
But it's not in the eyes, it's in the nose.
It's in the nose. Their predator nose.
They can like you can feel your nose getting warmer or something like that. Who knows? We don't have that sense, right? So the way they tested this was that they would have a couple of objects, one that was like slightly warmer than the other, and sort of detect the dog's ability to seek out the slightly warmer object.
Okay.
And they had to be trained, but then when they were trained, they had this capability, apparently.
Okay. And so fire beetles. Now, are they called fire beetles because they like fire, and therefore?
I don't know. It was just mentioned. This article's all about the dogs, but they're like,
Mostly.
Here are some other animals. It's not a
Okay.
common ability.
So
but
there's
here's some other
snakes
things.
out there that could find us and like.
Well, who knows? I mean, if you think about it, like if you're hunting at night and you're an animal that doesn't have amazing vision, like bats, right? I guess they use echolocation too. But you'd think that heat sensitivity would be such an obvious superpower to
every
detect
edge is an
living
edge
things.
when you're a predator.
Whoa, we should cross stitch that somewhere. Every edge is an edge, man.
Wow, that's fascinating.
So there you go.
Right.
Dogs
Well,
sniffing out
dogs.
hot smells.
Hot new smells just dropping for dogs.
What
That's
a life.
fascinating. Yeah.
What a life. I wouldn't want to be a dog.
You would not want to be a dog.
Mostly for this.
Mostly for the smell.
Hot, smelly garbage is all I smell all day.
You have a sensitive sense of smell, so you already have a tough time.
I'm a sensitive person to like criticism, but also
also to smell.
Stimuli.
Yeah, stimuli in any
sounds.
all senses.
We have this. I'm a very light sleeper,
Right.
and I have this story that I think best exemplifies how sensitive I am. I have a distinct memory of being woken up to the sound of a single dry leaf scraping across my sidewalk. That was enough to wake me up one day.
You're a light sleeper.
Well, that's my science
that's your fact. That's
Dogs
great.
Smelling hot, garbage.
Dogs smell hot.
That's all I have to say about that.
Why do you keep adding the garbage part?
Well, it's a hot, muggy day.
They're not smelling the garbage, they're smelling the heat.
I know, but they also have a nose that smells sensitive things. So a pile of hot garbage
is
would
doubly
be both
bad.
warm and stinky.
Okay,
yes.
I see. All right, well, I'm glad we learned that. Yep.
Aren't you happy to know that? Now, Vinny, I
all
want
right.
your fact.
You want my fact.
Yes,
so, all right, I'm gonna ask you, how do you feel about babies, Katie? Human babies.
Oh my God, adorable. We call them potatoes in this
potatoes,
house. We see a little baby, it's a little potato.
Yeah.
I love a baby.
How do you feel about babies in pain?
I hate it. Stop.
Yeah, we don't want that, right?
No.
We don't want that. Well, here's something that's interesting. There have been very few studies on baby pain.
Wow.
On how babies perceive and react to.
I can think of many reasons why no one would volunteer their baby to be tortured.
Yes, exactly.
And also babies can't talk about what they're feeling.
Well, here's the thing. Scientists have developed the infant pain scale.
Oh,
the newborn infant pain scale, NIPS. Now, I'm not making this up. So what happens is babies often have to undergo some testing when they're newborn, right? Now, sometimes you need to take blood from them. And the common way to do that is to do a little pinprick on the heel of their foot. Oh. They're little tiny babies. They just come into this world. And you got to take their blood. It's sad, but it has to happen to test for, whatever conditions may need to be tested for. And so you prick their heel. And then they cry and they're sad. No one would like it as an adult, but as a baby, you don't even know what's going on. And so they can tell like how much distress this baby is in using the infant pain scale. However, hot new research, this is good, is that playing music soothes the savage baby.
Savage baby?
Well, soothes the savage beast. I was riffing off of that common phrase, which I would now hope had been more common. So one of the interesting things is they have two cohorts of babies who were going to be tested anyways. One where they pinprick, got the blood, and didn't do anything. And then they did pinprick, got the blood, and then played some Mozart lullaby music. Some baby Einstein stuff for these babies. And it turns out, by the metrics developed on this infant pain scale, the babies felt less pain when there was music. They recovered more quickly from this incident when the music was playing.
And did the babies say why they think the music...
Well, the babies went on at length. Kind of had to cut them off at a certain point. But we don't know why. It's just there's something soothing about the music that maybe distracts them. I mean, we could hypothesize about what the mechanisms are. All we know is the result, which is that they are in distress for a shorter period of time.
I heard that this pain skill was largely developed for babies tripping and falling. Nip slips.
God. I did not see that coming, and I'm sad that I didn't.
Did they mention testing different types of music?
They only did this one study, and they wanted to control for music, so they just used this one set of music, this lullaby music.
How can a baby report pain?
I don't know. They must just, like, they said they'd check in on 1 minute intervals to see, like, whatever they're measuring for infant distress. Crying, squirming. I don't know what the metrics are here.
Yeah, because how could you even? I mean,
you could tell the difference between a newborn in distress and a calm baby, right?
Well, you're assuming crying is a negative thing. Maybe they're crying with joy.
Yeah. All right. I don't think you're gonna be allowed on this test study. One of the interesting things is that any previous testing, and not a lot of it has been done, has been exclusively done on white babies.
Oh, for Christ's sake.
This study went out of its way to make sure that that wasn't a thing. Just as so many studies test on men versus, completely ignoring the 51% of the population that is.
And white people.
Yeah. And so equally, a lot of studies tend to be done. If you're like, oh, we've got this university test. Oh, we're just going to sample from the people around you. Well, guess what? That sample is not going to be generally representative of the general population. So you end up with a white-favored bias of sampling when it comes to your test cohort. Oh. So this study went out of its way to not do that, which I appreciated.
But still tortured babies. All the babies.
I mean, they were going to get these heel pricks anyways. I'm assuming, unless they just went around and be like, hey, we're going to pinprick these babies for fun and science.
I couldn't do that.
I know, that's a tough one. That's a tough one, huh?
Can't you just tickle them and call it a day?
Well, it doesn't distress them in the same way. We have to study baby pain so we can avoid baby pain.
Yeah, I guess. It's a necessary evil.
It's a necessary evil.
I don't love it.
I don't love it either.
I feel bad for the babies.
Sorry, babies of the world.
Oh, little babies crying.
They didn't deserve it.
Well, good fact, though.
Good fact. Well, we're lessening
pain.
Lessening
yes.
pain.
Focus on the good. Focus on the good stuff.
Focus on the good stuff.
Okay, well, that's enough tomfoolery.
We did our science facts. Check.
Okay, so we did our
we did that, yeah. And then we did our facts.
What's
next? Well, guess what? I get to introduce our guest for today's episode.
Whoa.
Are you ready?
Yes.
Well, let's say hello to Asia Vighi. She's a PhD student at McGill University. She graduated with her bachelor's degree in bioengineering in 2022. Her current research focuses on transforming yeast cells into miniature factories that are capable of sustainably producing useful compounds for human use. In her free time, she enjoys reading fantasy books, making miniature rooms, and has recently gotten into ceramics. Let's say hello to Asia. Hey, Asia.
Hey.
Hi.
Welcome to the show. Thank you so much for joining us today.
Thanks for having me.
This is very, very exciting. So we're talking transgenic organisms, I'm assuming. Am I right? Well, what's
a transgenic?
Okay, well,
you jumped right ahead.
I saw transforming yeast cells. So let's break this down for our non-enlightened co-host. Let's talk about yeast. What the hell is a yeast, and why are we using it in this research?
So yeast, they're very cool organisms. So there's a lot of different species and strains that you can use. So different versions of the same organism, but they're similar to, we can think of bacteria. I think bacteria is maybe a little bit more well-known. It's single-celled, but where they essentially differ from bacteria is that they're a little bit more complex. So they're called eukaryotic organisms, so a little bit closer to a human cell, let's say. Mm-hmm. So although they're this independent single cell, that's quite simple and very well studied, very well developed in terms of lab techniques to work with them, they're also a little bit more of a closer approximation to higher organisms, so animals, and they are fungus, so mushrooms and all these various different things. But yeah, and specifically the yeast I use, actually, I think this is pretty cool, is brewer's yeast. So baker's yeast, it's the same yeast that's been used for, I think, thousands of years in fermentation, so in beer making, in bread making. So it's the same little guys doing some stuff for us nowadays.
Interesting. On that, the brewer's yeast, is it really like one species? Is it like a genus? There's a bunch of them that'll do it, or is it one species we use for all that?
So typically when we say brewer's yeast, we're referring to the species Saccharomyces cerevisiae, but in brewing in general, I think they've experimented with different organisms over the years, but this is the kind of typical budding yeast that people are referring to when they say yeast or brewer's yeast.
All right. And so you're using these yeast they're turning into factories to make things that humans can use. Is that what I'm understanding?
Yeah. So again, here there's I guess a lot to unpack and what I mean exactly by that. But I'd say the first thing to kind of consider is that naturally in the environment, there's a lot of different organisms that produce things that are super useful for us, right? Like I think most of our drugs are developed originally from plant-based compounds, so chemicals and active ingredients. And so there have been famous examples even within molds and fungi of this, right? Penicillin is one of them. I think it's maybe the first big example of this, where
my favorite mold byproduct.
Yeah, I think it's one that kind of revolutionized human history.
Top 5 molds.
Yeah. So naturally, organisms have these useful compounds, right? And so where transformation comes in is that when you're working with these simple organisms in particular, like bacteria and like yeast, what you can do is you can give them the genetic information, so the kind of recipes for building some of these compounds that they naturally can't produce. And so to do this, we usually do a process called transformation, which is when you give them this new DNA material that they don't already contain, so that they can then use that as an instruction booklet.
All right, so I'm thinking like Jurassic Park.
I think it's a little different, but I guess it's within the realm of genetically modified organisms, so I think that definitely falls within there.
So with transformation, when I think of what's going on with bacteria, typically you stick like a gene, like a recipe to make a cool thing, into a plasmid, which are like little circles of DNA, and then you convince the bacteria to take up, you give them, you don't talk to them, but you give them
Hello, bacteria, would you like some plasmids?
That's your charismatic voice. You give them the environment that's going to make them want to pick up stuff or be porous, and then they pick up the plasmid. And a lot of that works because bacteria in nature are trading plasmids like playing cards. There's mechanisms for them to do that naturally, and we sort of exploit that. When it comes to yeast, I know a lot less. Do they have a naturally occurring plasmid exchange system, or is that just a bacteria thing?
So the procedure is actually, it is, in terms of the general steps, it's almost identical to when you're working with bacteria. Yeast are just a little bit more sensitive, so they're a little bit more fragile to work with. You can kill the cells more easily.
Sounds like actors.
You do have to induce this competency in the cells. So use it through, there's different methods actually that you can use, but mainly I use a chemical-based method. So you give them this lithium acetate mixture, and the mixture of the chemicals that you're giving it essentially, open up the pores of the cells. It allows for the DNA to enter, very similar to when you do this with bacteria, but you do induce this competency. So naturally the yeast cells aren't just absorbing DNA or exchanging DNA from their environment. It's something that you induce into the cells. But yeah.
And so you're building these little packages. The yeast is like, oh, this is for me, and I take it in, and then it just like the little mechanisms inside are just going to generate whatever you tell it. You kind of like snuck it in there.
Yeah, and it's actually funny because so usually when you work with yeast, you work with bacteria, especially E. coli, a lot as well, because we kind of take advantage of the bacteria to produce the DNA for us, and then we actually extract it from the bacteria and then give it to the yeast cells.
Okay.
So usually you're doing a lot of bacterial transformation as well in the process, but just to produce these plasmids, these circular strands of DNA containing your genes. So actually, typically, when you're designing these experiments, the plasmids that you're creating, the circular strands of DNA contain these, we can call them convincing factors for both bacteria and yeast to take them up. So in theory, this does mean like if you're producing a protein, you could produce it in the bacteria as well. It's just some proteins. So these, just to give the basic definition of a protein, these kind of structures that are formed within the cell from the DNA, these little active structures, some of them are a little bit too complex for a lot of bacteria, which is why we use yeast.
So yeast is like a little bit better at making
it's a little bit more complex. Okay. Yeah, a little bit more like any animal or human cell. Right. Not so quite, but it's getting
it's like a level 2.
Yeah.
Well, because bacteria are prokaryotes, we're eukaryotes, and so we're a little different in how we handle
and the
DNA.
Yeast is a eukaryote.
Correct. So it's a double-edged sword, right? It has the abilities to recognize and deal with things that are close to us, but now you're dealing with a fancy actor. They're not as easy to
convince as like an E. coli. So we're using E. coli because it's like, oh, this is like baby talk, and then we're like, all right, we need to like have a book come out of this. So send it to the smart guy, which is yeast.
Basically, E. coli are it's just a lot easier to work with bacteria when you're working with making copies of DNA than with yeast.
Okay. And we still use bacteria for some things. So is it when we're trying to make a lot of, say, a protein that has like higher order structure, like proteins made of multiple polypeptides? Like what does it mean to be a complicated protein that requires or does better if we stick it in a yeast?
So the main reason I specifically use yeast is because the proteins I'm trying to produce will actually naturally localize themselves and fold correctly into their structures when they're located in a membrane or membrane-bound organelle. So in bacteria, we don't have this because they're prokaryotes. So this is why we need to work with yeast. So they associate to fats. We can think of the fats in the cell, and actually the compounds I'm producing as well. One of the main compounds I work with is a lipid. It's a fat. So when it's produced in the cell, it'll actually associate to this membrane. So it's important to have this membrane structure there. Other people will work with yeast as well because of certain post-translational modifications in the protein, but this gets more technical. And I think essentially the main idea is that they will only kind of form the correct structures in yeast in my particular case.
Yeah. And so the idea here is you've got a specific target, like a protein or molecule that you've got in mind, and you're like, all right, I need to make a ton of this stuff, and so I'm gonna give the instructions to this E. coli. This E. coli is gonna give me the instructions that a yeast can read. The yeast takes it and is like, I love this stuff, I'm gonna make a ton of it. And because it reproduces so quickly in such numbers that you end up with like a pretty easy way of making this target molecule. Is that what I'm understanding?
Yeah, and sometimes, optimizing it. So these compounds aren't necessarily always good for the yeast. Sometimes it can be toxic to the cell.
Sorry, yeast.
What is good about yeast and bacteria as well, right? We use bacteria in industry all the time, but it's really that you can scale up the production. So you can get these massive fermentation tanks, and this is almost an entirely different field of people who really specialize in that fermentation procedure, but they will really, really optimize it to kind of maximize the yield. So for each sugar you're putting in to feed the yeast, you're getting more and more of your product of interest. And what's cool too is that if you start to combine different research fields, there's a lot of people looking at using waste to feed the yeast. So you could take waste sugars and waste kind of biomass. Some people are looking at breaking down plastics and using the different carbons in there to feed the yeast. Whoa. So you can create these really circular economies with it. So that's one thing that's cool.
Harsh.
They're happy with just a little bit.
Are they happy? Do the NIPS test on the yeast.
Yeah, you know what? Capitalism ruins yeast too. No, but this is fascinating. So this is just this really cool way of like really getting, and so like these target, product of interest, as I think you called it. And so you've got this vat of fermenting yeast making a bunch of stuff, and it's like, all right, well, how do I get it out of, like, how do you retrieve it once you've got the yeast making the thing that you want it to make?
It actually, again, depends on what you're trying to make. Okay. So some people will use these microbes to produce proteins. So you can use proteins for therapy, like insulin, things like this as well. So in this case, after the yeast has grown for a certain period of time and produced enough of this protein, you can then extract it. So you'll break down the cells, you go through all these purification procedures, and then you'll eventually, get your your protein, and these can be used in therapies, and they work on this for gene therapies, vaccine production. In my case, actually, the compounds being produced, so mainly two different categories I work with. The big one for industry would be a group called carotenoids. So they're essentially, you may be familiar with beta carotene. It's in pretty much any multivitamin that you take. So carotenoids are essentially the compounds that make pretty much anything that's orange in the natural environment, including like the egg yolk and the chicken egg, or some of the taint in fish and shrimp comes from these compounds. So in the case of these compounds, they're not a protein. They can be purified afterwards if you're looking for one particular extract. But what's really nice is that for a lot of the applications we want to use the carotenoids, and we can use the whole cell. So you can just produce the yeast and then dry up this yeast and then just use the cells directly that already produce this compound.
So you don't even have to extract it in any way.
No, it depends on your purpose, but for this group of compounds, we don't need it to really be purified, so you can just kind of use it like that. And yeah, that's the main kind of approach in industry, but you can always break them down. So it depends where the compound's located. They're located in cell walls, so you would break them down, purify, and that's pretty much it.
Yeah. So the instructions for building the carotenoids are in the DNA?
Yeah, so...
I always think of DNA for being proteins, man. You're blowing my mind, Asia.
Because what the DNA has is the instructions to build the proteins that will make the compounds that we want.
Okay.
So that's essentially it. It's always the proteins doing the work, but in this case, our end goal isn't proteins; it's these compounds.
Fascinating.
Arcane Ninja RPG tells a story about ninjas and magic by mixing improv and role-playing. Meet our fledgling ninja and fall for them as they take their graduation exam and stumble upon unknown magic that will send them in an epic adventure. Root for them as the dice dictate just how successfully they face the threatening challenges that lay ahead. Binge on published episodes at the Pot Cavern Network or your podcast station of choice. Tune in every other Tuesday for your bi-weekly fix of Arcane Ninja RPG.
Thanks for listening.
So what kind of protein? I never think of proteins making things, but of course they do. Is it enzymes that are assembling?
They're
the
enzymes.
Okay.
All enzymes, yeah. So the enzymes that I introduce are naturally found in plants, mainly. And in plants, they're the enzymes that will complete these natural pathways and to make the pigments that give the coloration to the leaves or to the skin of a pumpkin, whatever it is. And then it's the DNA encoding for these enzymes that I'm putting in the yeast.
So you're adding the enzymes too?
Not directly. I'm adding these instructions to make the enzymes.
The DNA will make the enzymes. And then once the enzymes are there, they're like, let me make a carotenoid.
Okay.
They do their thing.
Yeah.
Okay.
And the reason this works too is because we are introducing enzymes in such a way that it's using naturally occurring pathways in the yeast.
Right, you don't have to do anything. They just, here's the instructions, here's the, I'm in
she's doing plenty. Let's not diminish Asia's contribution. You're doing nothing, Asia.
Not what I meant.
Real accusatory. So what's the point of this? Why do I want to make a bunch of carotenoid-filled cell membrane? Then what do you do? You give it to new species so that they are full of carotenoids and that's good for us? Or what do you do with them?
So for carotenoids, the main use is industrial in animal feed, and also I'm trying to produce one specific carotenoid, and this one also has applications as vitamins for humans. It's called lutein. It's helps with age-related macular degeneration, so it helps keep your eyesight good over time. The other compound I work with is a tanchinone, and these are chemotherapeutic compounds. They're anti-cancer compounds actually would be a better way to say it. So it could be used in treatment and the development of different medicines to treat cancers, specific cancers, not all of them. But the main reason we even, try to do this, I think a common question is why not just take it directly from the plants, right? If the plants can naturally produce it.
That was our next question.
Yeah. No, but it makes sense, right? It's like, why would you go through all this trouble? And the main reason, and I think especially when it comes to like these medicinal compounds, is that the way they're produced in plants, they're produced in something called a secondary metabolism. So it is not their primary central production mechanisms for the things that they need to grow. It's kind of like a side pathway. It's what makes all the signaling hormones in plants and things like this. And so they're produced in very, very small quantities, very, very small quantities. And so when we want to extract them from the plants, the procedure, I mean, it takes a huge amount of resources, a huge amount of time. It's very dependent on climate. It's dependent on harvest. A lot of times we can't produce these fully synthetically. We can't produce them chemically. So it can make the procedures very expensive in some cases, not in all, but in some cases. And it can make the procedure, very inaccessible to the general worldwide population. And so the idea with yeast is that it's kind of, first of all, if you can scale up the procedure enough, which it is very scalable, it reduces the cost, which makes it more accessible economically to people. And I think, especially when it comes to medicine, this is something that's super important to consider.
Yeah.
Cheap.
And it's better for the environment too, usually.
Right. You're not taking up all this land.
Yeah.
So carotenoids, correct me, I'm probably wrong here. There's a link somewhere between carotenoids and vitamin A. Am I correct? Beta carotene and vitamin A, it's a type of carotenoid or something. So like, I know why we need carotenoids. Why does a plant need carotenoids? They ain't got no eyes.
Yeah, so plants need carotenoids. So some algae produce it as well. So microalgae, most of them act as a sort of, in relation to the pigment. So you can think of like chlorophyll giving these green kind of coloration to the leaves. They're kind of acting as a secondary pigment. So a lot of it is to do with, and it depends exactly on which one you're looking at, but to do with absorption of light, which is important for photosynthetic organisms. But a lot of carotenoids, and even for humans, one of the reasons, and animals in general, one of the reasons they're super important is because they are antioxidants. And so in this kind of light activated pathway, even in plants, you're producing a lot of these oxygen byproducts that are highly reactive. It's not the oxygen that we need kind of to breathe. It's a different form of oxygen that has these free electrons. Yeah, and so these antioxidants will help counteract and balance out the effects of these pro-oxidants that are produced in these pathways. So this is one of the natural, evolutionary reasons for it. This is not my area of expertise, but it's similar to, when the sunlight starts changing in the fall and the green pigmentation dies off and the leaves are all orange, it's these carotenoids becoming visible. They're always there, but now they're becoming visible because they do have this important role as well in the leaves and in photosynthesis. Yeah.
Now, can we use this method that you're using with yeast to make anything, or what are the limits right now here?
Oh, this field is a very rapidly growing one. In some ways, there are no limits. There's a few big groups now. Like if you look, NASA has this space food challenge.
Okay.
And this is like a big example of it. If you just Google NASA space food challenge, I think you'll see, yeast comes up as one of them, because yeast is also being looked at as a way to produce alternatives to dairy, alternatives to meat, as well as just like this sugar. I don't know if we would want to just eat yeast, but maybe to enhance the nutritional value of certain
like
things.
Yeast oatmeal.
Nutritional yeast.
Ugh.
Yeah, so yeast is really, really being looked at to produce a lot of the foods of the future, if you will, in quotation marks.
Right.
And space foods. I think the idea there too is just this idea of accessibility, because really, as long as you can build a tank that'll grow the yeast, you can produce whatever it is that your yeast has been. Yeah. Wow. And there aren't really any limitations. There's like laboratory scientific limitations. Some things are harder than others to produce, but in theory, with time, I think most things can be made.
Like an amazing organic 3D printer.
Well, yeah, kind of. And I think that's what's really cool about using these microbes, is because they have this natural capacity to convert sugars into whatever else an organism needs. So
now, Asia, I love food. I get a lot of pleasure from food. I'm scared of this future you're working towards. We're eating bowls of dry yeast. How do we stop you? You evil genius. How do we stop you?
I have no intention on eating eating yeast every day for the rest of my life. So
that's the world you want. Admit it.
Big
it
yeast.
has a very particular smell. I smell it enough on a daily basis.
Ooh,
I would rather not.
Oh,
it just smells
tough.
like rotting bread a little bit.
Fermentation.
It's a very like
cheese.
Acidic bread.
Acidic bread.
Yeah, it's not great.
Yeah, okay. I'm not sold on it yet for food.
So, as you know, Asia, in CEGEP, we do a little bit of transformation. And I mean, I'm an ecologist. This is not my area. But, the steps seem to be, you got the gene for the thing you want. I mean, you had to make it to begin with. So we're sort of jumping in mid-process. You stick it in a plasmid. You stick that plasmid in the yeast grows and replicates and makes a bunch of it. What's like the biggest challenge in that rough framework, or an additional step that I omitted? Like, what's like the real clincher for you as a scientist? Like, oh, this part is so hard to get to be successful.
So assuming you have your DNA sequence and you haven't accidentally introduced like a random mutation, and, assuming you haven't contaminated your samples, because this is very easy, right? So a bacteria is a contaminant to yeast. So typically when I'm working on a yeast transformation, so bacteria, a lot of times we work around just the Bunsen burner, because the Bunsen burner will create the sterile, depends the bacteria you're working with, but E. coli creates a sterile area around the flame. But when you're working with yeast, you in theory could use the Bunsen burner, but we often use a biosafety cabinet. So it's kind of like a fume hood, but just optimized to keep, you're not, the air is filtered in a different way, because it's microbes that you're working with in there. So I think the biggest challenge is human error a lot of times, in the sense that it is super easy to introduce bacteria into your samples. But also yeast cells they're not necessarily super welcoming to the DNA that you're putting in. So you do have to, open the walls up and shock them with some heat so that, they'll open, and then make sure that they close and that the cells recover. But at the same time are very fragile. So it's super easy to kill your cells when you're working with them. Not quite as easy as mammalian cells. I will say that, way easier.
Oh, really?
Yeah, no, way easier. This is not, but they're challenging, but in general, the transformation process is something that's been pretty optimized over time. So this is not typically the barrier. It can be, but it's not the limiting kind of step in the process. It's more the growth and the production and making sure your DNA sequences are correct and things like that.
So, I mean, maybe one day we'll make a yeast that could eliminate the human error in this process.
Whoa.
We have a yeast train to make more yeast stuff. That's the future.
That's what you want? That's the future you want, Vinny?
No, I don't. So, okay. So this sounds like really exciting, like an amazing way to produce a whole variety of things that we can use. What would be something like, oh man, if we could make this, this would be like top of the list to make.
The Mount Everest.
Like, is it carotenoids or beta carotene, or is it like, man, if I could make, I don't know, aspirin.
I think it's mainly, I'm not sure of the exact name of the study, but quite some time ago, there was this massive study that was conducted on plants, looking at every single compound in a bunch of different plant species, what is useful for humans. And from this study, they found a lot of different compounds that were super effective in treating, I think, cancer is a big one, but not only cancers, but cancer is a big category that a lot of money has been invested into finding various cures for cancer. Yeah, so from this study, they essentially found a number of these agents that would be super effective in treating these different cancers. And so one of them is Taxol. This one is quite well known, I believe, for breast cancer, but don't quote me on that. But yeah, so essentially, I think these are the ones that really people are looking at because of the fact that they're produced in such low quantities in plants. I mean, you need like numerous trees just to get 1 milligram. So it's not feasible. It's extremely expensive and just not sustainable in any way. Even if you were someone who didn't care about like the forest itself, it's not, something that can be scaled up to actually help any significant number of people. So I think these are really, really where the field focuses a lot of efforts. Yeah.
Right. Well, if you could do it in a vat, then you're golden.
Yeah, it'd be great. And that's the problem, is that traditionally a lot of time, like chemical synthesis, right, is used, but because some of these compounds are just so complex and so specific that one bond just attached in the wrong orientation makes a completely different compound. And so that's why using these natural organisms is just easier sometimes. Yeah.
Cool.
Now, I know a tiny bit about bacteria's sort of autoimmune system for dealing with the things that infect them, which are viruses. And we exploit some of that to do this technology. But what do yeasts have a natural autoimmune system that you need to suppress? Like when you're trying to stick something in a yeast, do you have to suppress something to keep it from fighting it?
So we're not so much suppressing. So one thing that is interesting, actually, an interesting difference between working with bacteria and yeast is that when you're working with bacteria, typically you'll take advantage of antibiotic resistance to kind of convince the bacteria to take up these genes. So what you do is you add the gene encoding for the resistance, something that will produce resistance to an antibiotic, and then you grow the bacteria in the antibiotic with antibiotic in their growth culture. So only the bacteria that have taken up that gene alongside a gene of interest usually will be able to grow. In yeast, we don't really use this mechanism. You can. There are some antibiotics, fungicides, I guess, that you can use, but they're not as selective because yeast are more complex. They do have a lot more kind of defense mechanisms in place if you were to just add an antibiotic and things like that in their media. So what we actually take advantage of are the amino acids. So these are the essential amino acids that any organism needs to survive. What we do is the yeast we work with in the lab is actually incapable. We've engineered it to be incapable of producing some of these essential ones, and then we don't add it to their growth media, but we add the gene encoding for how to produce them alongside our gene of interest. So that's typically the approach we take. So it's called auxotrophic selection. It's a deficiency, in the growth media. Yeah.
Got him.
Auxotrophic again.
We tricked them.
So how'd you get into this, Asia? How'd you get into the biotech and stuff?
That's a really good question. So as part of my undergrad, it was, a degree requirement. I had to do a research project and a capstone. So I'm in engineering. A capstone just means this 8-month kind of group design project. At the time, my PI, my supervisor, she was a newer professor in the department. So she was setting up her lab. And no one was really doing research in this generally referred to as synthetic biology, this kind of field. And so, I started researching, the courses started popping up and they were new and I was curious. So I reached out to her. I did a summer research project with her and eventually the capstone. And then she said, hey, do you want to stay on? I think you're doing a good job. And I said, yeah, you know what? Actually, I would like to stay on. So this is how I kind of ended up here.
Very cool.
It's something that always just really fascinated me. It's this capacity of natural organisms to just make stuff. It's just really cool. Yeah.
That is very, very cool.
Absolutely.
And were you, as a kid, were you like, oh, I love building things? Or were you naturally engineering inclined? Or was this something you discovered later on?
I've had many passions in my life. When I was much younger, I was convinced I would go into classical music. I was one of those 7-year-old kids. My mom took me to Juilliard at the time. I would have never gotten into Juilliard, but this was my, big goal. So I was my mother's quite artistic, so I think that's just naturally where I kind of got pushed. But I used to always do these little kits with my dad. We'd build model airplanes and we would build these, they have those little STEM, build a little robot kits.
Yes.
Right.
So that's how it kind of started. And so one day I was trying to decide what I wanted to go into in CEGEP. I was there and I was kind of just okay, what do I like? And I liked biology and I liked math.
Yeah.
Because bioengineering as a whole, the undergrad, it can be a lot of different things. I mean, some of my friends are building, robots for surgery. It's quite diverse in terms of, as a field. It's very multidisciplinary, and that's also what I liked. And I think that is maybe the part of my character that carried through. It's this desire to do something that isn't necessarily one specific thing. It's kind of you get to explore many different things. So yeah.
That's amazing.
And even in your off time, you're building.
Yes.
You said this is one of your hobbies, building miniature rooms. We had to get through all the transformation talk to get here. I need to hear more about what you're building and what tiny mice. Are they for yeast? How miniature are these rooms?
They're not that miniature. So I I don't know what got me into this. It may have been TikToks. I do like having a little arts and craft hobby on the side. And so it started with these kits that you could buy to make miniature coffee shops or miniature greenhouses. And then it kind of went out of control from there.
Are we talking book nooks or
Not
like
quite.
doll houses? Okay.
I wouldn't call them a dollhouse. I wouldn't call them these little books. They're kind of an in-between. Like, you're just making, like, the very first kit I got was like a miniature greenhouse and flower shop.
Wow.
And so from there, this weird thing started happening where then every little piece of trash I had, I was like, oh, this could be a flower pot, or this could be like, a broom. So I just have this box of essentially trash that every once in a while I pull out and I make a little something for one of my little various.
Is this?
Yeah.
I'm hoping that after the recording you will show one to us because I'm dying to see this. I've gotten a bit into book nooking and I get it.
Yeah.
It's very satisfying, the gluing of tiny things.
Yeah. And I have given some of the ones I made to my dad just to keep because I could keep them in my apartment. But a lot of times it's just like little things. Like I'll make a little flower pot and then I made this little fruit cup with little strawberries in it the other day. But it's just little things like this.
This sounds amazing.
Yeah.
I love this. I first thought of miniature making from the TV show The Wire. Where a detective very famously he was bored at work, at least at the beginning of the series, build like doll furniture. And he made a remark that like that's how he makes most of his money. And just like this tiny, tiny little
that
detailed
stuff
things.
Goes for a lot. If you want to buy it on Etsy or something, it's pretty pricey. I always consider, I think I'd consider selling some on the side, but for now,
you will need to tell us if you're making yeast-sized yogurt fruit cups.
Or we just train the yeast to do it for us.
Oh my gosh!
But then we would deprive Asia of the joy of
making
it. Yeah.
Well, Asia, this has been amazing.
Yes.
Thank you for taking the time away from the smelly acid bread yeast to tell us about your research.
Oh, it's been a pleasure. Thanks for having me.
Thank you.
Keep fighting the good fight.
Yeah. Wow, this is really cool.
Have you heard of transformation, Vinny?
This is totally new to me. Yeah. I mean, it makes total sense that nature is making stuff all the time, right? Like, it's just, you can't stop it. And if we can kind of direct it to make stuff that we humans need, and this way of, just generating a lot of it in a really efficient way, why not? How great is that? What a great idea.
Absolutely. Way better for the environment, economically, making these things far more affordable.
And these stuff that we need, like these medicines that must be super rare, but maybe effective in humans. Yeah, why don't we just get yeast to make it? Just kick it over to them and give them a little sugar and kick out some medicine.
You don't even need to give them the good sugar. Trash.
That's right.
Trash. Turning trash into treasure. That's yeast.
That's yeast. Thank you, yeast.
Thank you, yeast. This episode's dedicated to yeast.
I think that's it for us today.
We learned a lot. We learned a lot about the thing you talked about.
Yeah, babies.
We learned a lot about babies.
Little babies said.
The NIPS test.
The NIPS test, yes.
And dog noses. Smelling out
heat.
Stink garbage.
Detecting hotties.
And transformation yeast.
Transformation yeast, yeah.
All right, Vinny, why don't you send them out on our socials?
Well, if you're interested in following us, you can check us out at LRG Pod on Facebook, Instagram, TikTok, and on YouTube. You can just search us. We're not LRG Pod on YouTube, unfortunately.
Well, look up.
We couldn't get them all. But Learn Real Good Podcast on YouTube, you'll find us. And if you know somebody who would be interested in being a guest on our podcast, if you're into STEM, in the grad student in the STEM fields, just send us an email at learnrealgoodpodcast@gmail.com. That's learnrealgoodpodcast@gmail.com.
Well, that's it for us this time. Thank you, Vinny.
Thank you, Katie.
Thank you, Asia.
Amazing.
Thank you for listening.
Thank you, Yeast.
Thank you, Yeast. Thank you, Dog Noses.
Yes.
Thank you, everyone.
Babies.
See you next time.
Bye-bye.
Bye-bye.